In September of 2007, a European research satellite called Phylum M3 returned from 10 days in low Earth orbit. Among the passengers were a group of water bears. The vacuum of space had not killed them. The tardigrade is a microscopic animal of its own phylum, Tardigrada. Around 1,300 species carry formal scientific descriptions. A typical adult measures between 0.3 and 0.5 of a millimeter long, around 1/100th of an inch. 8 legs, each tipped with 4 to 8 claws, a simple brain. A through gut, a body plan that has stayed recognizable for 500 million years. They live almost everywhere, in glacier meltwater, in abyssal deep marine sediment, in Antarctic ice, in the moss on a suburban wall. They do not require extreme environments, they tolerate extremes when they occur. The interesting fact about a tardigrade is not where it lives. The interesting fact is what happens when the water leaves. Under desiccation, or freezing, or osmotic stress, the animal performs a particular sequence. It retracts its legs and head. It expels nearly every bit of its body water, retaining, in the most measured species, between 2 and 3% of what an active tardigrade holds. It collapses to about 1/10 of its ordinary volume. It enters a state, named nearly 2 centuries ago, called the ton. In the ton, no measurable metabolism is occurring. By most definitions, the animal is not alive. But it is also not dead. Rehydrated in a drop of water, it resumes movement within minutes to hours. The phenomenon has a name: cryptobiosis, latent life. That dates to the 20th century, though the behavior itself appeared in the 1770s notebooks of the Italian naturalist Lazzaro Spallanzani, who gave the group its name. What the ton can survive is the property that makes tardigrades interesting. Temperatures within a few degrees of absolute zero and above 150 degrees Celsius, above the boiling point of water by a wide margin. Pressures up to around 6,000 atmospheres, roughly 6 times the pressure at the deepest point in the ocean. Ionizing radiation at doses 1,000 times the lethal dose for any vertebrate. And, as the 2007 Phylum M3 experiment demonstrated, the vacuum of space itself. The Phylum M3 experiment was led by Ingemar Jönsson, a Swedish evolutionary biologist at Kristianstad University. He and his colleagues flew 2 species of tardigrade, Milnesium tardigradum and Richtersius coronifer, on the outside of the satellite for 10 days, fully exposed to space. Vacuum alone, most survived and laid eggs after return. Vacuum plus full-spectrum solar ultraviolet radiation, survival dropped sharply, but not to zero. The result was reported in Current Biology in 2008. It was the first time any animal had been shown to survive unshielded exposure to outer space. The mechanism behind this tolerance was not worked out until 2017. Thomas Boothby and his co-authors, publishing in Molecular Cell, showed that tardigrades produce a class of proteins found nowhere else in nature, tardigrade-specific intrinsically disordered proteins. As the animal loses water, these proteins do not fold up or crystallize, they vitrify. They form an amorphous glass-like solid inside the cell, locking every structure, membranes, DNA, proteins in place stable until water returns. The animal is held suspended in a glassy matrix of its own making. Disable the genes that encode these proteins, and the animal's desiccation tolerance collapses. The longest documented cryptobiotic survival for any tardigrade came from Antarctica. In 1983, a moss sample was collected at Showa Station on the Soya coast and stored frozen at -20 degrees Celsius. In 2014, a team led by Megumi Tsujimoto at the National Institute of Polar Research retrieved the sample, rehydrated it, and recovered 2 living individuals of Acutuncus antarcticus and a separate egg. The longer-lived individual laid 19 eggs, 14 of them hatched. The animal had been in cryptobiosis for 30.5 years. Writing in Cryobiology in 2016, Tsujimoto noted that the recovery time for the first egg laid was almost twice the median. The animal took longer to reach reproductive function, but reached it. The implications of that single observation, and of the proteins behind it are still being worked through. Laboratories have begun using tardigrade proteins to stabilize vaccines and enzymes at room temperature. Astrobiologists have re-examined what can be called habitable. The question of what it means for something to be alive between the cells of a dry, mossy sample on a shelf has been quietly reopened. A tardigrade is not difficult to kill when it is active. A drop of seawater will kill a freshwater species in minutes. But when the water leaves and the glass forms and the animal suspends itself, it waits. We are not quite sure during that time what it is. We are fairly sure what it is not. And in the right drop of water, at the right moment, it will start moving again.